All right. Well, hello everyone. What I'd like to do is spend a little time reviewing and preparing for the test. So I've got the test here. What I'm going to do is take a look at the test and then just jot down concepts that appear on the test. So let's start first with lacrimal glands. So if you have your eye, here's an eye, sitting up above the eye is the lacrimal gland, and then the lacrimal gland will produce tears, and then tears will streak across the eye and help clean the eye. And then you have these little canaliculi that lead to this lacrimal duct, and the tears drain away and then down to your pharynx to the back of your throat. Okay, so the lacrimal gland produces the tears to help clean your eye. They drain away and then travel down your throat, and you swallow your tears. In terms of the levels of the eye or layers of structure of the eye, if we're imagining an eyeball here, we have three layers that we're going to pay attention to. The outermost layer is the sclera. The sclera is the white part of the eye. And then around the front of the eye, that sclera becomes a clear dome, and that is the cornea. Now, the middle layer of the eye is the choroid layer. And the choroid layer has blood vessels to supply nutrients and pigments to help absorb the extra wavelengths of light. And then the innermost layer of the eye is, oh boy, that's similar color. That's the retina. And the retina has rods and cones to help absorb the wavelengths of light. So three layers of the eye to know. The sclera is the white part of the eye. That's tough and protective. The choroid is the middle layer of the eye, and that has blood vessels and pigment. And then the retina is the innermost layer, has the rods and cones to absorb light energy. Let's see. Now, in the front of the eye, we have, if I redraw the eye here, yeah, we take the eye and the pupil is that hole in the front of the eye, and then the iris is the circular muscle around that. Okay. Now, the pupil can dilate or constrict sort of based on what the eye needs. So again, the pupil is the dark spot in front of the eye. Now, if you need more light to come in, then the iris will relax and the pupil will dilate. And then say if it's really bright, then the iris will constrict and the pupil will get narrower. So if it's like a dimly lit situation, the iris will relax and the pupil will dilate so you can allow more light into the eye. And then if it's really bright, the iris can constrict and the pupil will narrow and you allow less light into the eye. Let's see. Now, one thing we know about the front of the eye, the cornea here, is that it's covered in a membrane called the conjunctiva. The conjunctiva is this clear membrane over the front of the eye and on the inside of the eyelids that produces this lubricating fluid so that you can blink and you're fine. Now, conjunctivitis is when you have an infection here and you have inflammation in that spot. All right. When we look at the retina, there is an area in the back of the retina that is referred to as the fovea centralis. The fovea centralis is important because that is where we focus, if here's the lens, the fovea centralis is where we focus the light in our eyes. The fovea centralis is all cones and it's packed with cones, so it's where we have our sharpest vision. Let's see. The rods tend to be more distributed in our peripheral vision, and they're good for detecting motion. And then we concentrate a bunch of cones in the fovea centralis, so we see best under bright light conditions. Let's see. Now, we've looked at corneal transplants. If we think of an eye, again, the cornea is that dome on the front of the eye, and the cornea can become damaged. But because there are no blood vessels in the cornea, it doesn't have as big a rejection issue. So you can actually do corneal transplants, and they will cut out the cornea from one person and from a cadaver, and then can place it on a person. So you can get those corneal transplants. Let's see. Now, the retina in the back of the eye here, the retina has the rods and cones. So if we can imagine a retina here with the rods and cones, if part of the retina was to become detached, then what would happen is the retina could fold over and then it would block part of the flow of light, and then you would lose part of your field of vision. So a detached retina is problematic because, again, I've compared it to like a poster folding over. If the poster folds over, then you're not going to be able to see well. You're going to block part of your field of vision. All right. Let's see. Now, the iris, this colorful part of the eye, it contains pigments that absorb light energy. So if you have a more darkly pigmented eye, that means less light energy strikes the back of the eye. If you have more lightly colored eyes, then more light energy strikes the back of the eye. So the pigment in the iris is important because what that pigment does is absorb excess light energy, because you don't want too much light energy striking the retina, otherwise it would be damaging. Let's see. Now, one thing that happens in the back of the eye is we have an area called the optic disc. The optic disc is significant because it's where all the rods and cones have their axons leave the back of the eye. So the optic disc is this spot that has no photoreceptors, so it is our blind spot. So all the wires next to the back of the eye, and then you have the optic nerve that'll travel to the back of your brain. Let's see. Now, one thing that can happen is because the chamber in the front of the eye here, the anterior chamber, gets fluid from aqueous humor to provide nutrients for the cornea, the ciliary body will produce that aqueous humor, and then this canal of Schlemm will drain it away from the eye. If you overproduce fluid or block up this drain, then pressure can build in the eye, and then the pressure can damage the rods and cones in the back of the eye. If you damage those rods and cones, then that could affect a person's ability to see. So that's exactly what glaucoma does. With glaucoma, you have increased pressure in the front of the eye, and then that increased pressure in the eye damages rods and cones, and then the person loses part of his or her vision. Okay, now, with our eyes, we can focus light energy, right? You can look at something that is, up close. So I don't know, like a miniature person. And what can happen is if you're looking at something up close, the lens in your eye can bulge. And if the lens bulges, then the image will focus on your eye. So the key here is that the lens can change its shape to focus the light. One thing that happens is as you get older, the lens doesn't bend as easily. And if the lens doesn't bend as easily, then the image focuses further back and things look blurry. So this is called presbyopia. Presby means elder. So with presbyopia, because the lens hardens, it doesn't bend as much, so the light focuses behind the eye, and so what you see is blurry. So that's why people may need readers as they get older. It's so that the lenses on a pair of reading glasses can bend that light more substantially and get the light to focus on the retina. All right. If you recall, when you look at, a tongue, the bumps on the tongue are not taste buds. The bumps on the tongue are actually papillae. So if we imagine looking at this from the side, Boy, this is not drawn particularly well. If you can imagine looking at this on the side, one of those bumps is a papilla. And so here is one of those bumps. Here's the papilla. Now, down within the bumps are the taste buds. And within the taste buds are the taste receptors. Okay? So you can have receptors for the various tastes within those taste buds. Again, I compare the papilla to an apartment complex, and the taste buds are like the individual apartments in the entire apartment complex. And just like different people live in different apartments, you can have different combinations of taste receptors living in those taste buds. So again, think of a taste bud as being the apartment. The papilla, or the bump on the tongue, is the apartment building, and then the receptors are the people that live in the apartments. Now, smell or vision is detecting light. Smell and taste are both chemical senses, so you're detecting chemicals. And then hearing is sensing mechanical force, so you're sensing vibrations traveling through the air. So that's one thing to keep in mind. Related to that, flavor is comprised of multiple factors. You can have tastes or different tastes. You can have the way things smell. You can have temperature, and you can have texture affect all of those things. So think about the lab activity that you did where you pinched your nose shut. If you reduce your sense of smell, that's going to affect the flavor. Or if you smell one thing while you taste something else, that's going to affect the flavor. If you add different tastes in, like you add, salt, something sour, something bitter, something umami, that's going to change the flavor. Even changing the texture of the food can alter that experience of flavor. All right, things to know. Make sure you review the path of light through the eye. Also, make sure you know the path of energy through the ear. You have the auricle or pinna, then the acoustic meatus or the ear canal, then you have the middle ear with the hammer, anvil, and stirrup. Then you have the cochlea and the endolymph within that cochlea. And then the waves of energy are detected in the organ of Corti. So you can just sort of review that stuff. Make sure you know the path of light through the eye and the path of energy through the ear. Thinking of the middle ear, you've got those three bones, the malleus, the incus, and then, gosh, I'm terrible at drawing this stuff, the stapes. So hammer, anvil, and stirrup. And when your tympanic membrane vibrates, that vibrates the hammer, which vibrates the anvil, which vibrates the stirrup. The stirrup pushes on the oval window, and that vibrates. And then that sends vibrations through the fluid in the cochlea. And then inside the cochlea, you have that spiral organ of Corti. You have the basilar membrane. Embedded in that are the hair cells. And then sitting on top of that is the tectorial membrane. So the idea is, sound comes in through your auricle or pinna, through the auditory canal or your ear canal. It strikes the tympanic membrane. That makes the hammer, anvil, and stirrup vibrate. That causes the oval window to vibrate. Energy is sent through the endolymph in the cochlea. That makes the basilar membrane vibrate. And then those hair cells bump up against the tectorial membrane. The hair cells bend and send action potentials. to the brain, and then you hear things. Let's see. Now, if we think about the inner ear, you've got a few different areas here. One part of the inner ear is the cochlea. That's the snail-looking part. That is for hearing. The next portion is the vestibule. The vestibule is sort of an entry area. That is for your static equilibrium. Again, that is sort of sensing the tilt of your head. And then you have those semicircular canals. And those are for sensing your dynamic equilibrium or the motion of your head. Okay. Let's see. Let me jump back to this image here. This structure here inside the cochlea, this is the organ of Corti. So this is the actual organ of hearing. It's got the basilar membrane, the hair cells, tectorial membrane. Again, waves of energy traveling through the endolymph make the basilar membrane rock. The hair cells bump up against the tectorial membrane and bend, and that sends out action potentials. So the organ of Corti is your structure of hearing. Let's see. Now, your middle ear, one funky thing about your middle ear is that it's a little air-filled chamber, right? The hammer and anvil syrup are in this air-filled chamber. Well, this is the pharyngotympanic tube. That pharyngotympanic tube, it leads to your pharynx, or the back of your throat. So if you get a nasal infection, a throat infection, cold, anything like that, the infection can travel up that pharyngotympanic tube and cause otitis media or a middle ear infection. And again, remember, little kids, their pharyngotympanic tube tends to be more horizontal, so the mucus doesn't drain as easily, so they are prone to middle ear infections. Adults have more vertical pharyngotympanic tubes, so the mucus drains away, and they don't get those sorts of issues as readily. Let's see. All right, let's talk about the vestibule here. If we think about the vestibule, what we have is this membrane. We have hair cells. And then the hair cells are covered in a gel. And then that gel is covered in these little crystals or otoliths. If you tilt your head, you will change the angle of that membrane. That causes the otoliths and gel to sag or pull. And then that makes the hairs on the hair cells bend. And then they fire off an action potential, and you sense the tilt of your head. Not the movement, but the tilt of your head. Let's see. So again, static equilibrium is about the position of your head. Dynamic equilibrium is about the movement of your head. Let's see. Conduction deafness versus sensory neural deafness. With conduction deafness, what you're doing is blocking the sound waves. So like here, you have your auricle or your pinna, the fleshy part of the ear, or the ear canal, and then you move to the middle ear. With conduction deafness, you're blocking those sound waves. With sensory neural deafness, you've got problems with your organ of Corti or your cochlear nerve. So sensory neural is wiring issues. Conduction deafness is blocking sound waves. Let's see. So vision is our ability to see, olfaction is our ability to smell, and gustation is our ability to taste. So those are things to consider there. Let's see. We talked about ceruminous glands. Ceruminous glands. Oops. Ceruminous glands produce cerumen, that's earwax, and that traps junk that gets into your auditory canal. So it basically captures and covers up anything that gets inside. The tympanic membrane is your eardrum. That is a structure that vibrates to send on vibrations to your middle ear. Your auditory tube. That's the same thing as your ear canal, which is the same thing as your external acoustic meatus, all same terms. Let's see, otoliths. Those are the little crystals in the vestibule that help you sense the tilt of your head. Let's see, the cochlea. It's the snail-looking part of your inner ear, and it's used for hearing. Let's see, the organ of Corti. That is the structure in the ear that is related to sensing hearing. That's in your cochlea. And then the semicircular canals are the portions of your inner ear that sense the dynamic equilibrium of the motion of your head. Let's see, one other thing to make sure you know, know a diagram. Oops, if I can spell diagram. Know your diagram of the eye and a diagram of the ear. Okay, so be familiar with the diagram of the eye and a diagram of the ear. You did the eye diagram assignment, and then the ear diagram, you could look in your textbook or the front of that lab packet would be good as well. Okay, other concepts to finish up with here. We talked about knowing the path of light and the path of sound. Sort of know what's going on there. Be familiar with flavor and how it's produced and how you can alter your ability to taste. And be familiar with the relationship between static and dynamic equilibrium. All right, so that covers the concepts that are on the test. As you review stuff, if you have any questions, let me know. Otherwise, I wish you the best of luck and hope you do well. Thank you much.